Good morning, everyone. So now I'm going to discuss with you my next video with you. It is on antimicrobial resistance transmission. And before I start my video, I want to request you, if this video appears useful to you and if it is helpful to you, kindly subscribe to my YouTube channel. That is Dr. Nitika Pharmacology Discussion. And now I'm going to discuss with you on the topic antimicrobial resistance transmission. Now, antimicrobial agents, they get resistant to bacteria, and these bacteria transmit the antimicrobial resistance from one bacteria to another. And antimicrobial resistance can be of two types. That is, first is vertical, and second is horizontal. Vertical transmission is from the parent drug to the progeny. Means the bacteria divide, multiply regularly. The parent drug divide and multiply, and the progeny is formed. And thus, there is a vertical transmission of the antimicrobial resistance from the parent to the progeny. And the mechanisms of action of the antimicrobial resistance of the vertical transmission can be of five types. First is enzymes, inactivating enzymes developed by the bacteria. There are certain bacteria that develop inactivating enzymes which inactivate the drug in the bacteria. So these can be of which inactivate the drug by releasing inactivating enzymes. These can be of three types: A, B, C. A is aminoglycoside, B is beta-lactam, C is chloramphenicol. And beta-lactams antibiotics, they develop the alternating enzymes, that is the inactivating enzymes, that is beta-lactamases. Next is the altered target. What is the altered target? Altered target is the bacteria, they develop the altered target to the drug. Means the drug doesn't bind to the original target, it binds to the altered target, so resistance develops. These can be in the case of MRSA and VRSA. In the MRSA, that is methicillin resistant Staph aureus bacteria, they develop protein binding, penicillin binding proteins. PBPs are developed, that is altered target sites are developed. Penicillin binding proteins in case of MRSA, and BRSA also develop altered target sites. And third is the decrease in the permeability. Means the permeability of the drug into the bacteria decreases, and this can be permeability of the bacteria to the drug decreases, and this can be seen with aminoglycosides. Now, fourth is efflux pumps. The bacteria, they develop efflux pumps which efflux the drug from inside of the bacteria to the outside. Tetracyclines are the drugs which develop resistance to the efflux pumps. And newer drug of the tetracycline group is tigecycline, which was resistant to this efflux pumps. And now the bacteria has also developed the efflux pumps against tigecycline. Fifth is the altered metabolism. That is, the metabolism of the bacteria is altered, leading to death of the bacteria. So the bacteria, the folic acid metabolism is disrupted by the sulfonamides. They inhibit the folic acid synthase enzyme. So folic acid, newer folic acid is not produced, and they start using the preformed folic acid. Bacteria start using the preformed folic acid, so the resistance develops. Now, this was all about the vertical transmission. Now, next is the horizontal transmission. That is from one bacteria to the another. Means that is from the brother to sister. That is one bacteria, the resistance develops to the another bacteria. It can be of three types. That is conjugation, transduction, and transformation. Now, conjugation is the type of the antimicrobial resistance in which there's bacteria which develops the resistance genes inside its body. These resistance genes, they multiply inside the one bacteria, and they are responsible for resistance of the bacteria. Now, the one bacteria, it develops the physical contact with the other bacteria, And this physical contact from between one bacteria and the other bacteria is known as the sex pili. And the resistance genes get transferred from one bacteria to the another bacteria with the help of the sex pili. And this is the most common method of the horizontal transmission. This is the resistance genes gets transferred from one bacteria to the another bacteria by the sex pili. That is the conjugation. Next is this transduction. Transduction is when there is a virus which infects the bacteria. Means there is a resistance gene present inside the bacteria which is multiplying. The virus infects the bacteria, and this virus which infects the bacteria is known as the bacteriophage. It attaches to the surface of the bacteria, resistant bacteria, and resistance genes get incorporated into the bacteriophage. And then this bacteriophage moves to the another bacteria and the transfers the resistance gene to the another bacteria, leading to resistance of the other bacteria also. This is the transduction. The next is the transformation. It is that the one bacteria which is producing the a lot of number of resistance genes, and it is a like plasmid or plasmid or transposon. These they are resistance genes are produced by the bacteria, leading to resistance of that bacteria. It releases the resistance gene. The bacteria releases resistance genes just like the free environmental DNA, and this free environmental DNA is taken up by the another bacteria. And this free environmental DNA process of the resistance gene transmission is known as the transformation. This is the least common method of the horizontal transmission of the antimicrobial resistance. So, I have discussed the vertical and horizontal antimicrobial resistance transmission. I hope you understood my video. And if you like this video and if it is helpful to you, kindly subscribe to my YouTube channel. This is Dr. Nitika Pharmacology Discussion. Thank you. Be happy, be healthy, be blessed. Thank you. Happy learning.